Hello and welcome to another video in the lecture series entitled Advanced Biomaterials. In the previous video, we had looked into polymeric nanoparticles and we had discussed what are polymeric nanoparticles and how they can be used as delivery vehicles for the transport or for the delivering of genes or different therapeutic agents such as drugs inside a target cell or target tissue. So we will also see why such polymeric constructs are essential in preventing the degradation of the active analyte or the biological component inside biological systems. We have seen how these constructs are fabricated in the two different aspects of these systems, and also we have seen about some of the important considerations or criterions which are to be kept into consideration while trying to build such polymeric systems or nanosystems for delivering of different drugs or genes for therapeutic purpose. So in today's video, we will be looking at the mechanism in which polymeric nanoparticles can be used to deliver genes inside the nucleus of a target cell. So we will be looking at the polymeric nanoparticles for intracellular delivery of DNA. For the intracellular delivery of DNA. Now, this mechanism is interesting, or it is important to know in order to fabricate, in order to construct these effective delivery vehicles for gene delivery inside target tissues or cells. So, first consideration is the combination of these polymeric nanoparticles with the DNA or nucleic acids to form polyplexes. So, the first step is complexation of anionic DNA with cationic polymers to form polyplexes. So basically, if this is a polymeric chain or a polymeric network, in fabrication of polymeric nanoparticles, these will assume a spherical shape. And these are positively charged. And the nucleic acids, which will be in the form of DNA, will be negatively charged. The phosphate groups of this DNA will be negatively charged. So these DNA will be inside these polymeric systems, and electrostatic interactions will hold or stabilize this structure inside the polymeric systems. So the outermost layer or the shell, if we draw a simplified diagram, will have these positive charges, and inside will be the DNA, which will have the negative charges, and positive negative electrostatic interaction will take place to stabilize these particles. So these are the polyplexes. Now, the second step will be let me erase out this part so we can write it here. Will be the cellular uptake of the polyplexes via the different endocytic pathways or endocytic routes. So the cellular uptake of polyplexes via endocytosis. So these polymeric nanoparticles or the polyplexes in combination with this DNA will actually enter the cell through the process of endocytosis. So this is the cell membrane which will endocytose the polymeric nanoparticles. So this will be the whole cell. And these polymeric nanoparticles will enter the cells via endocytosis. Now, once these polymeric nanoparticles are inside the cells through this membrane invagination systems, these nanoparticles will be engulfed or taken up by the endosomal complex. So the third step will be of this mechanism will be the enclosure and subsequent release of the polyplexes from endolysosomal compartments. So this will be the enclosure of, and subsequent it will be after enclosure, then these particles will be modified and released from these endosomal complexes. The polyplexes will be released from the endosomal complexes to give rise to the fourth step, which is the nucleic acid and polymeric remnants which will enter the nucleus of the cell. So herein in this endosome it will undergo some type of processing so that these complexes will be modified and will the active in the target gene or the nucleic acid or DNA in this case will enter the nucleus of the cell, which is the target area. So the fourth step will be the release of free DNA from the polyplexes leaving behind the polymeric remnants. Now, these polymeric remnants, as we had discussed, these are biodegradable. So, these will be taken up by the cells and these will be degraded inside the cells. And since these materials are biocompatible with no or with insignificant cytotoxicities associated with them, the degradation products will not cause any harm whatsoever to the cells. Whereas this target nucleic acid or target DNA will then be transported or carried into the nucleus, which is the target site of action. So, the fifth step of this process would be the transfer of DNA into the nucleus for expression by the different nuclear membrane transport proteins. So the transfer of DNA into the nucleus for its expression by the different nuclear membrane transport proteins. So in the nuclear membrane, there will be these membrane transport proteins which will help in uptaking the DNA from the cytosol inside the nucleus through the nuclear membrane, and inside the nucleus these DNA will then cause the expression of the specific gene or specific protein of interest. So, expression of this DNA or the gene will take place. Now, we have seen that how these polyplexes can be used to deliver the gene or the DNA inside the nucleus of a target cell to promote or to favor the expression of that particular gene of interest. Now, in this process, these polyplexes actually, or the DNA or the target or the active bioactive ingredient will encounter certain barriers in this system or in this pathway from the production, from their in the entire physiological process in which it is injected and till it reaches its final site of action. So, we will discuss some of these barriers. So, let me erase out some of this part. So, we will look at some of the barriers which are encountered during this process. So, barriers encountered during the process of polyplex mediated gene delivery inside nucleus. So, the first barrier is the different proteins or the different enzymes which are present in the blood. So, different components in the blood. So, in the human blood, it contains different enzymes and different proteins which can actually cleave or which can actually degrade these polyplexes. So, in order to save these polyplexes from the blood, from the different enzymes present in the blood, then these can be modified accordingly. Right? So, the second barrier which is encountered is the plasma membrane. The plasma membrane is a phospholipid bilayer, and in many cases it has been found that in order to actively introduce the gene or the target analyte inside the cell or inside the nucleus, it has to first cross the plasma membrane. So in order to cross the plasma membrane, it is done usually in case of polymeric nanoparticles, it is through endocytosis, and therefore modifications need to be made to promote or to influence this process of endocytosis. The third barrier, and one of the most significant barriers which needs to be considered while engineering such delivery vehicles, is the endosome. So this endosome can actually degrade these polymeric nanoparticles, and in order to protect from these degrading enzymes which are present inside the endosomes, so these endosomes, one is it fuses with the lysosomes, it becomes an endolysosome, and these lysosomes will have hydrolytic enzymes which can degrade the polymeric scaffold, the polymeric matrix, and once it reaches the DNA, it will degrade the DNA, and therefore our delivery vehicles will become ineffective. So in order to protect this from the endosome, it has to be escaped from the endosome. So endosomal escape is an essential consideration while designing such polyplex systems. So we need to make such systems so that it can escape from the endosomes without damaging the DNA or keeping the DNA intact. So endosomal escape. Is an essential consideration. Finally, the fourth consideration or the fourth barrier is the nuclear membrane. Again, the system or the gene of interest should have this particular size, should have this particular characteristic, such that it can be recognized by the nuclear membrane transport proteins and effectively introduce, or it can effectively selectively enter the nucleus of the cells to promote its expression. So these are some of the important barriers which needs to be identified and which needs to be considered while engineering such polyplex systems for delivering or for targeting the DNA or gene of interest inside the nucleus for a specific expression. So with this, we'll end today's video here. In the next videos or in the upcoming videos, we'll look at some of the general preparation techniques of polymeric nanoparticles. So hope you enjoyed it. See you in the next video. Thank you.